Hello, everyone. Welcome to Backyard Musings, broadcasting live from Apple Valley. Again, it's a happy new year. Thank you for joining us. I'm Steve.
I'm Scott.
All
Happy
right.
New Year.
Happy New Year. All right. Researchers at the CUNY Graduate Center have made a groundbreaking discovery in Alzheimer's disease research.
We just talked about that.
Yeah. Identifying a critical link between cellular stress in the brain and disease progression. Their study focuses on microglia, the brain's immune cells, which play dual roles in either protecting or harming brain health. Don't know why we want to mess with it if it protects and harms. If you take out how do you separate the harmful from the protection ones?
I don't know. Hopefully they figured out a way to
You
do
got to
that.
tweak that, man.
Yeah.
By targeting harmful microglia through specific pathways, this research opens new avenues to potentially reversing Alzheimer's symptoms and providing hope for effective treatments. Would you like to know more? Nod your head up and down.
Researchers with the Advanced Science Research Center at the CUNY Graduate Center have unveiled a critical mechanism that links cellular stress in the brain to the progression of Alzheimer's disease. The study, published in the journal Neuron, highlights microglia, the brain's primary immune cells, as central players in both the protective and harmful responses associated with the disease. Microglia,
How to pronounce it?
yeah, I don't know. Microglia,
Julia.
often dubbed the brain's first responders, are now recognized as a significant causal cell type in Alzheimer's pathology. However, these cells play a double-edged role. Some protect brain health, and while others worsen neurodegeneration.
Mm-hmm.
Understanding the functional differences between these microglia populations has been a research focus for Pinar Ayata, the study's principal investigator and a professor with the CUNY ASRC Neuroscience Initiative and the CUNY Graduate Center's Biology and Biochemistry programs.
He's got some titles and some degrees.
Sounds like he's a busy guy.
Yeah.
We're hoping that's a guy, right? That's the guy?
That's the guy.
Okay.
That's the guy. Quote, we set out to answer what are the harmful microglia in Alzheimer's disease and how can we therapeutically target them, said Ayata. We pinpointed a novel neurodegenerative microglia phenotype in Alzheimer's disease characterized by a stress-related signaling pathway. Oh, that's a mouthful right there. You really gotta rewind and listen to us what we said, because there's a lot there. The research team discovered that activation of the stress pathway, known as the integrated stress response, prompts microglia to produce and release toxic lipids. These lipids damage neurons in oligodendrocytes. Let me spell it out here. Let me sound it out. Oligodendrocyte progenitor cells. Oligodendrocyte progenitor cells. Two cell types essentially for brain function and most impacted in Alzheimer's disease. Everybody knows this. Right, those two. Right.
If they don't, they do now.
Right. Blocking this stress response or the lipid synthesis pathway reverse symptoms of Alzheimer's disease in preclinical models. That's promising. That's pretty cool.
Yes. Key findings. Dark microglia and Alzheimer's disease. Using electron microscopy, the researchers identified an accumulation of dark microglia, a subset of microglia associated with cellular stress and neurodegeneration in postmortem brain tissues from Alzheimer's patients. These cells were present at twice the levels seen in the healthy aged individuals.
Ooh.
Toxic lipid secretion. The ISR pathway in the microglia was shown to drive the synthesis and release of harmful lipids that contribute to synapse loss. Is that right?
Synapse loss.
Synapse loss.
That's the fire with the brains, where they fire off and they connect.
Okay. A hallmark of Alzheimer's disease. Therapeutic potential. In mouse models,
we love mice. We love mice.
They're giving their life for this thing.
Yeah.
Inhibiting ISR activation or lipid synthesis prevented synapse loss, and accumulation of neurodegenerative tau proteins. We talked about tau stuff the other day.
Yep.
Offering a promise. Oh, when we were talking about
Yeah.
this,
I said we had a
this
segment
topic.
coming up. Yeah.
Yep. Offering a promising pathway for therapeutic intervention.
I wonder if there's if we could find any, put herrings and mice together, for like research.
Herring?
Yeah, we just did a segment on herrings.
Yeah, but how would you test them in the labs? How would you communicate
With water. How do you communicate with mice?
Well, you watch them running around in their cage.
Well, then you watch the herring swim around.
Right?
Let's use them.
Yeah, I guess.
Let's see the mice of the sea, right?
I guess.
Okay.
I'm good with the mice and monkeys. And what did
Monkey
you say?
Island.
What's the other thing we don't like?
Monkey.
Beagles.
Yeah.
We don't want testing on beagles.
No testing on beagles. They're cute. Quote, these findings reveal a critical link between cellular stress and the neurotoxic effects of microglia and Alzheimer's disease, said the study's co-lead author, Anna Fleury, a member of IATA's lab and a PhD student with the CUNY Graduate Center's biology program. Targeting this pathway may open up new avenues for treatment by either halting the toxic lipid production or preventing the activation of harmful microglial phenotypes. Okay.
Yeah. This research underscores the potential of developing drugs that target specific microglia populations or their stress-induced mechanisms. Quote, such treatments could significantly slow or even reverse the progression of Alzheimer's disease, which is good.
Yeah.
Offering hope to millions of patients and their families. Absolutely. Explained co-lead author Lean, Al-Jayusi. Al-Jayusi.
Got it.
A member of IATA's lab and a PhD student with the CUNY Graduate Center biology program. The study represents a major leap forward in understanding the cellular underpinnings of Alzheimer's and emphasizes the importance of microglia health in maintaining overall brain function.
Okay,
so there you have it.
We talked about a lot of gobbledygook in there, big words.
A lot of work. Yeah, some of them weren't even that big, but they're
cutting edge, and it's working towards a cure to Alzheimer's. That's
this is exciting. This is a good,
the words are not
good. Yeah, no,
but the research is.
Yeah, good stuff. Definitely good stuff.
All right, thanks, folks. We'll catch you on the next one.
Take care. Happy New Year.